Hi everyone, today we're going to look into a little bit of IB biology, and this topic that we're going to specifically dive into is nucleic acids, and we're going to cover it until topic 1.2.3, which is sugar-phosphate bonding and sugar-phosphate backbone of DNA and RNA. So let's get started. Right, first, two things that we have to know about the function of nucleic acids. Nucleic acids generally serve two functions, which is first, passing information between generations, and also second, coding for protein production. And there are two types of nucleic acids, DNA, which is deoxyribose nucleic acids, and RNA, ribonucleic acids. In terms of looking at a single nucleotide, the general structure of an RNA and a DNA nucleotide is generally the same, but they do have minor differences, which I'll explain later on. But in terms of the general composition, they all have three elements, which is pentose sugar. In the case of RNA, this is ribose sugar, and in the case of DNA, this is deoxyribose. And we also have a nitrogen-containing organic base and a phosphate group. So the general sort of structure of a nucleotide is as follows, as you can see in this diagram right here. We have a phosphate group, and the phosphate group is acidic and it's negatively charged. And we have a pentose sugar. And then we also have a nitrogen-containing base. And the sort of bonding that forms between the phosphate group and the sugar, the pentose sugar, is covalent. So is the bonding between the pentose sugar and the nitrogenous base. But even though they're both covalent bonds, they're different bonds. This I will explain later on, but in terms of the bonding that forms right here, this is phosphodiester bonding, and this is glycosidic bonding. And these bonds that form between one single nucleotide and the antiparallel nucleotide are hydrogen bonds. This I'll explain further later on. We're first going to look at nitrogenous bases. And in terms of the bases, we have 5 types of bases, which is adenine, thymine, cytosine, guanine, and uracil. But uracil, this one's only used when we're talking about RNA nucleotides, so you don't need to consider uracil when talking about DNA nucleotides. And these nitrogen-containing organic bases have either 1 or 2 rings of carbon. And what I mean by that is we classify these nitrogen-containing organic bases that have 2 rings as purine bases, and you can kind of see it right here. Purine bases, they have 4, 6, and 5 membered rings, so it has a total of 2 rings. But these guys, cytosine and thymine and uracil, they're pyrimidine bases, and they generally have only 1 ring composed of 6 membered rings. Yes. Right, and this is the part that I was talking about earlier on in terms of the bonding that forms between each of these components. Between the phosphate group and the pentose sugar, we get a phosphodiester bond, and between the pentose sugar and the nitrogenous base, we get a glycosidic bond. And let's dive in further to what these bonds each look like. In terms of the phosphodiester bond, we need to dive into the definition of what an ester bond is. An ester bond is when the OH group of the carboxylic acid and the H group of an alcohol condense together in order to form water and is removed. That's when a bonding forms, and that sort of bonding forms between the phosphate group and the sugar, forming a phosphodiester bond. The bond that forms between the sugar and the nitrogenous base is called a glycosidic bond, and this is between different carbohydrate molecules, and this is again another condensation reaction. Right, given that we understand the sorts of bondings that are involved within a single nucleotide, let's try to move on to the difference between an RNA and a DNA nucleotide. So the main difference between an RNA and a DNA nucleotide is that in carbon 2, RNA has an OH group, but DNA attached to carbon 2 has only an H group. And this is exactly why the pentose sugar in an RNA nucleotide is called the ribose sugar, but then in a DNA nucleotide, it's deoxyribose, given the absence of oxygen. Right, now let's try to move on to sugar-phosphate bonding, and more specifically, how the sort of sugar-phosphate backbone is created when we get a nucleotide strand. So what happens when we get a nucleotide strand is one nucleotide is connected or bonded to another nucleotide with the formation of a phosphodiester bond. So what happens is, with carbon 3 of the pentose sugar of one nucleotide, the other phosphate group of another nucleotide kind of bonds through a phosphodiester bond. So phosphodiester bonds are, again, condensation reactions. So what happens is, say that this is the sort of phosphate group of another nucleotide, and this is the pentose sugar of one nucleotide. What happens is, from this OH group that we mentioned earlier on, and from the OH group of the phosphate group, the OH of the phosphate and the H of the OH group attached to carbon 3 of the pentose sugar in one nucleotide, they are removed in the form of water in a condensation reaction of the phosphodiester bond. And so we get a phosphodiester bond right here, and therefore we get these nucleotides kind of connecting together. And they all connect together to form this sort of strand.